Dr. Parmar. She'll be teaching a class during our summer taste, and she'll share a little bit about that. But today is about our graduates from the Masters in Biotechnology. So with that being said, Dr. Parmar, take it away.
Well, thank you so much. Good morning, everyone. And this is Dr. Nitika Parmar, Professor of Biology and also an instructor for the past, I think, 15 years for OLI. Some of you may already know me and some of you may not. But it's a great pleasure and honor to be here and presenting this talk. This talk is actually focusing on stem cells and regenerative medicine. And towards the end, after I've done my talk for about 30 minutes, which will be pretty much just the background information about stem cells, what they are, what they can do, then I will pass the baton to my wonderful students. These are graduate students in the MS Biotech program at Channel Islands. And they have divided themselves into 3 groups. And each group will be focusing on a very specific human disease that has great potential to be sort of, I wouldn't use the word cure right now, but that has great potential to be addressed by the use of stem cells. So that's where you will get a very fine taste about the power of stem cells. That's what the students will be presenting. But without further ado, I'm going to do my presentation. And if I can request all of you, the audience, to please hold off on your questions till the end of the presentation, that would be so much appreciated. And I'm more than happy to answer all your questions towards the end. So please bear with me as I share my screen. Okay, can everyone see this? I just need to know, can everyone see this? Just a show of hand.
You're good.
Yes.
Perfect. All right, so as I mentioned here, this talk is on stem cells. And several of you may have read some very old time magazines which focused on the power of cloning and essentially what happens when sort of like synthetic human beings are made by the genetic engineering of the genome. The genome is basically the DNA inside our body. And now we have the power to actually manipulate that DNA in whatever manner we want to and make the changes, engineer it, and put it back in a human body. We have the power, and to make things even more dangerous, the technology is actually pretty simple. And there are certain individuals who are actually doing this in their garage. I read a book that somebody is actually doing this in the garage, which is absolutely outrageous. But thankfully, there is a ban on reproductive cloning and reproductive engineering, which prevents individuals from ordering their babies based on specific design, based on specific looks, hair color, eye color, skin color, even IQ levels. So thankfully, reproductive cloning is banned as of now in humans, but reproductive cloning is not banned in animals. And so animals have been cloned for the past 22 years. We have cloned dogs, cloned cats, sheep, goats, buffaloes, you name it. But we don't have human cloning allowed right now, which is very, very important that it should not be allowed because it can be misused extensively. But we have what we call therapeutic cloning, which is being done in humans, particularly those who have very detrimental lifelong diseases because of certain changes in their DNA. And therapeutic cloning allows that fault in the DNA to be fixed. And eventually, whatever is missing in the human's body, essentially, that can actually lead to the treatment of a disease. So that's where the golden lining is for the use of stem cells. So there's a whole lot of debate about this, and the debate never ends. And it's all tied to stem cells. Are stem cells actually a blessing, or are they to the contrary? And a lot of people, both scientists and non-scientists, are still arguing whether we should use stem cells or not. But let me tell you, 95% of the scientists in the world today advocate for the use of stem cells for the betterment of human society. When I say betterment, I'm not talking about looks, I'm talking about disease fixation, basically for therapeutic purposes. So let's get into some nitty-gritty background here, since we've heard of the word stem cell quite often now. The word stem simply means fundamental line from which other cells and other kinds of tissues have arisen. So stem means something very basic, something which is coming like from the origin. So that's why we use the word stem cell. So what exactly is a stem cell? Stem cells are very, very unique. Now, if you look at cells in your body, such as the skin cells or hair cells or cells in your stomach, they are extremely specialized. And when I say specialized, they have been designed in such a manner that they can only perform very specific functions. So the skin cell cannot digest your food. And likewise, the stomach cells cannot grow hair because each one of them has its own functionality. And this is where the stem cell is very different from all the other cells in the body because of two primary features. Number one, they have an unlimited potential to renew themselves, which means they can divide forever, provided we give them the right food, the right house, the right gases, the right light. They have this capacity to self-renew, which means they can copy themselves again and again and again. And this is very important because these cells are very important to fix any damage which is happening in our body. Anytime we have minor injuries inside the body, which we are not even aware of, these cells come into play, they quickly divide and basically regenerate the tissue, which is going on day and night in our bodies, but it's at a very minor level. And then the second major feature of stem cell, which I think makes them extremely attractive, is the capacity to differentiate into different kinds of cells. Now, the differentiation word may be a bit technical, so let me make this easy. So stem cell is very unique because if you provide them the right proteins, the right food, they can grow into any cell type that you want. They can become a brain cell, they can become a pancreatic cell, they can become a kidney cell, they can become an eye cell, and you name it. So that is why the power of stem cell is huge, because you can manipulate how you want to convert the stem cell into whatever tissue you want it to turn, and then sort of like use that tissue for regenerative or therapeutic purposes. So like I mentioned, these stem cells are actually unspecialized. They're not specialized, which means they are not further down in their lineage. They are very, very early cells in the body. They're very primordial cells in the body. And like I said, they can be induced, they can be coaxed, they can be encouraged under the right conditions to become cells which have different kinds of potencies. So this is why these two features make the stem cells extremely powerful for fixing diseases. So this is just a pictorial just to show you what happens. You start with one stem cell and it divides and divides and divides, and you maintain a stem cell pool in the body. And this pool is extremely critical, like I mentioned. Whenever there is a break in a blood capillary in a body, you need to fix it right away. And that's where the stem cell comes. They turn themselves into blood cells and they fix the break in the body. And all this is going on regularly in our body. We're not aware of it. These are things which are going on constantly in our body. And then they can differentiate. And in the body, this actually goes on naturally because that specialization is extremely critical to either get rid of dirty, old, damaged cells and replace them with new cells. Because what happens is if we allow the damaged cells to consist
Dr. Palmer, can you hear me? Yes, Dr. Palmer went frozen. Thank you, Kirby. We will see what we can do to get her back. Give us 1 second. We lost her. All right, she'll be back. This is the beauty of Zoom, how it just like, and technology, that's the negative of technology. It can kick you out when you don't want to be kicked out. So please bear with us. She'll be right back.
I think I got disconnected for some reason.
You did for 1 second.
Yeah.
You got about 6 sentences that we stopped hearing you. So
6 seconds?
6 sentences probably.
Okay, great. I was at this slide, so I'm going to continue with this slide where I stopped. So like I mentioned, the word potency means what is the strength of something or what's the power of something. And with regard to stem cells, we classify the stem cells into 3 different types: pluripotent, multipotent, and unipotent. And it's very easy to understand. Uni means one, multi means several, and pluri means everything. So a pluripotent stem cell is the best. It is the most important cell in the body because it can specialize into any cell type. And this is basically what happens in an embryo. When a sperm meets with an egg and there is fertilization, it leads to the formation of an embryo. And in the cells, which are part of the embryo, they are all pluripotent because the embryo has to turn into a complete human being with eyes, nose, ears, hair, all the different organ systems of the body. So embryonic stem cells are pluripotent. They can specialize into any cell type in the body. And then the second kind is the multipotent. Multi means they can actually specialize into different kinds of cells, but not all kinds of cells in the body. And one example of a multipotent stem cell is a blood cell, especially those in the bone marrow. So the stem cells in the bone marrow can differentiate into both RBCs, the red blood cells, WBCs, the white blood cells, as well as the platelets. So they can only turn into blood cell types. They cannot turn into neurons or skin cells. So that's why we use the word multipotent. And then the unipotent is the one which has the minimum potency because that can only produce one cell type, not more than one. So that's why the word uni is being used here. All right. So what are the various kinds of stem cells? I'm sure several of you are aware here. The one that is talked about extensively is the embryonic stem cells. And as the name suggests, these stem cells are coming from the embryo. And these are sort of the most valuable cells in the body because they give rise to an entirely new human being. And then the second one is the adult stem cells. As the name suggests, these stem cells are not coming from the embryo. These are normally present in our bodies, in everyone's body. You, me, everyone, each and every human being, even the animals have adult stem cells. And the reason why we have stem cells is because we need them for tissue repair and tissue regeneration every time as we are getting old or we undergo some kind of injuries in the body. So adult stem cells are extremely critical for our health. Then the third kind, which is sort of used and not used specifically in certain countries, is the cord blood stem cells. As the name implies, cord blood is actually coming from the placenta. At the time of delivery, the placenta is harvested and the stem cells are actually derived from them. And finally, the stem cells which are surrounding the baby as the fetus is growing inside a mother's womb, there is a fluid around that growing embryo, so to speak. And that fluid is actually providing a cushioning effect to the baby as the baby grows. And that is known as amniotic fluid or AF. And the stem cells which are derived from AF are known as amniotic fluid stem cells. So 4 different types, but like I said here, embryonic stem cells are pluripotent, adult stem cells and cord blood stem cells are multipotent, and amniotic could be unipotent or multipotent. So this is a pictorial I wanted to show of a 4 to 5 day old embryo. And at that stage in our scientific language, we call that embryo a blastocyst. So please don't worry about the scientific nomenclature I'm using. This is simply a 4-day-old embryo growing inside a woman's body. So where do we find the embryonic stem cells? These orange cells that you're seeing right here, these are the embryonic stem cells, which are surrounded by a layer of supportive cells known as trophoblast. But it's only the orange cells which are embryonic stem cells, not the blue ones. The blue ones are just supportive in nature. The orange ones have a capacity to specialize into any kind of cell type in the body, and that's how an embryo grows. So this is one of my coolest and my favorite pictures. This is a real electronic micrograph of a growing blastocyst. This is 4-cell stage embryo, which becomes, this is an early blastocyst, then a late version of the blastocyst, which moves on to the 4th stage, and finally it goes into the morula stage. So you can see the cells right here inside. This is known as the inner cell mass, and that's where the embryonic stem cells are harvested from. But these days, the embryos are not destroyed. Nobody actually harvests embryonic stem cells anymore. They used to do it, I would say, roughly in 2003, about 20 years ago, because at that time we didn't have much knowledge about another kind of stem cells known as induced pluripotent stem cell, which I'll talk about in a minute. So at that time, the embryo was the only source to harvest these stem cells. And the embryos were collected from fertilization clinics, from abortions and so forth, which we'll discuss just a few slides down. So what are the challenges? So I may be making it sound really, really simple, but there are certain challenges. Even let's say hypothetically, we do grow the embryonic stem cells in the lab in a little house, which is known as a petri dish. To make them turn into different kinds of cells in the body is not an easy job. I mean, it's really easy to say, for me to say, okay, let's turn it into skin, let's turn it into brain cells or the blood cells or the liver or intestine or kidney. But it's not that easy, because the food that we have to provide to the embryonic stem cells for them to be converted into these different cell types is very, very different. What food works for neurons will not work for skin, and likewise. So maybe the blood food likes pizza and fries, maybe the neurons like broccoli and spinach, maybe the skin likes, donuts and cookies. So you see, the point I'm making is here, the growth conditions under which these embryonic stem cells turn into specialized cells is very, very different from each other. And it is extremely important for scientists like us in the lab to fine-tune these growth conditions. Otherwise, we will not be able to turn those embryonic stem cells into the various cell types. So this is where a lot of research and development is going on in different scientists' lab to find out what proteins, basically we use genes, what genes need to be turned on to convert these embryonic stem cells into these different kinds. So, but we've come a long way thanks to funding, $1 million funding that we received. So we've come a long way in determining what food is good for these ES cells. So that's one of the challenges. Now let's move on to the second type, which is the adult stem cells. Like I mentioned here, these are normally present in the human body, and their main function is to maintain our tissues and engage in repair as the human body is growing old, or we fall sick, or we have a fall and we have a damage inside the body. So this is where all the adult stem cells come into play, and they're found pretty much in every organ in the body. You name it. I have a picture here that shows you where they are all found. They are found in the brain, they're found in the liver, and of course, most important, blood vessels, because we may not realize it, but we are constantly tearing blood vessels inside the body almost every day, very, very minute capillaries. We bump our shoulder somewhere, there's a little break or a blood capillary inside the body. We need to fix it right away. That's where the stem cells come. We have them in the pancreas, the skin, the bone marrow is very, very important for cancer patients, because the bone marrow actually gets destroyed upon chemotherapy, and that's where the bone marrow transplantation is extremely important to regenerate the bone marrow so the person can actually develop immunity towards future diseases. So there's lots of stem cells, adult stem cells in the bone marrow. We have it in the stomach too, in the muscle, lungs, and very important in the eye. in the eye is very important. And that's a great area which is being researched by my colleague in UC Santa Barbara. His name is Dr. Dennis Clegg. He is trying to explore how to transplant retinal stem cells in people who have macular degeneration or near blindness and restore their sight back. So that's really, really cool research that he's doing there. So plenty of stem cells. The embryonic come from the embryo, the blastocyst, while the adult stem cells can come from different parts of the body, as well as the amniotic fluid and the placenta. So we have lots of resources, but again, having said that, the maximum number of cells are present in the embryo, and the least number of cells are present in the amniotic fluid or the placenta. It's just about 0.5%, whereas the adults are in different populations in different organ systems. Right, so adult tissue stem cells, what can they do essentially? Well, like I said, they're used for a variety of purposes. They can be differentiated into different kinds of multipotent stem cells, in which case they can turn into blood cells. And it's extremely critical that these multipotent stem cells have the full capacity to differentiate into what we call the blood lineage, because we don't want them all to be turning into RBCs and not becoming WBCs or platelets. That really will not serve the purpose. We need them to turn into all these four kinds of cells: WBCs, RBCs, macrophages, platelets, and so forth. So these are the multipotent stem cells. And then we have various questions, because now adult stem cells are actively being explored for the past about 12 years, but there are still so many unanswered questions. I've listed some of the questions here. Where are they present? Are they present in each and every tissue on the body? How many kinds are there? And what is their source? How do they divide? And are they very flexible? Can they be turned into any kind of cell? And most importantly, as a scientist, the question that really intrigues me is, what kind of food do we need to supply to them so they can immediately migrate to sites of disease and injury and start fixing the damaged tissue? So still a lot of questions unanswered, but there's lots of research going on in this area. The third kind of stem cell is the cord blood stem cells. And as the name suggests, these are derived from the placenta or the umbilical cord. And they're mostly multipotent in nature because they can only give rise to stem cells. This is blood stem cells. Hematopoietic means they have a capacity to turn into WBCs, RBCs, and all the different kinds of blood cells. They can be triggered to become brain cells, but that trigger is very, very tricky. So it's not very easy to turn cord blood cells, stem cells, into neurons. So basically, this is what happens today. I know several of my students, they have told me personally, as they gave birth to their young ones, they actually chose to save and preserve the umbilical cord, which is shown right here. And this is filled with hematopoietic stem cells. And the reason why the umbilical cord or the placenta is being preserved today is just in case in a subsequent pregnancy, if there is a genetic defect in their following babies, they can quickly harvest these stem cells from the placenta and put them in the new baby's body and fix the disease if the disease were to arise down the line. So several women I know personally are more and more choosing to actually preserve their placenta for future purposes. And then the last one is the one derived from amniotic fluid. Like I mentioned, this is the fluid which is surrounding the baby as the fetus is actually growing. And so you can see there's a needle that goes into the amniotic fluid. That is simply to harvest some DNA from the baby to see if the baby has genetic defects. But this is the fluid which is harvested to isolate the stem cells. But amniotic fluid does not have very many stem cells. In fact, they only have 0.4 to 0.6% stem cell. So not a rich source of stem cells, unlike the adult stem cell tissues. So going back to the whole ethical question, which used to be a big, big challenge earlier when embryos were utilized for isolating the embryonic stem cells was, where do we get the embryos from? And mostly the embryos were obtained from fertility clinics, post in vitro fertilization trials, where a couple had maybe 9 embryos, but they chose to implant maybe 2 or 3. So what do you do with the remaining 5 or 6 embryos? And they don't want any more implantations. Well, with the proper consent of the hospital as well as the clinic and the parents, these leftover embryos were obtained and embryonic stem cells were harvested from them, and essentially the cell lines were grown. And sometimes it happens that certain embryos are not implanted into the woman's body after IVF trials because they have found there are certain genetic defects in the growing fetus, as a result of which those embryos are not deemed healthy. And even in those cases, those embryonic stem cells, which may have genetic defects, but they can still serve as excellent sources for scientific purposes, for research. So why don't we use adult stem cells more often? Well, the only reason is, unlike the embryonic stem cells, which are pluripotent and very, very flexible, adult stem cells are not very flexible. They tend to be multipotent, not pluripotent, as a result of which we cannot have them to specialize into any kind of cell type in the body. But we have actually bypassed that challenge now. And I'll tell you in a minute what that challenge was and how we bypassed it. So the ethical debate actually continues. Proponents say that we must use all the frozen embryos which are lying in a fertility clinic freezer. And if a couple chooses not to implant them and they don't want any more babies, then what are we going to do with these hundreds and thousands of frozen embryos? So the proponents of embryonic stem cell research say that anyway, these embryos are going to be destroyed eventually, so why not use them for scientific purposes? While the opponents say, well, the embryos are life, so they should not be destroyed needlessly, because somebody who does not have children or is not capable of having children, these embryos can actually be given to a childless or an infertile couple, and these embryos can be used for adoption. So this debate sort of like continues, but it's not as active as it was about 20 years ago, because now hardly anybody is using embryonic stem cells because of the discovery of what we call iPSCs. So what do we do with these stem cells? Well, loads and loads of things, and my students will actually talk about it. We can use them for testing new drugs. We can use them for studying how mechanisms occur, how these cells talk to each other, how do they decide when they want to change into something, so basically developmental protocols. And most importantly, we could use them for therapeutic purposes, which we call cell-based therapies. And they can be used for sort of addressing diseases such as diabetes, Parkinson's, Alzheimer's, and so forth, and spinal cord injury also, which we will discuss, my students will discuss shortly. All right, so stem cells, in order for stem cells to function inside the body, it's very, very important that they be able to perform their job really, really well, which means they should be able to divide extensively. They should not halt their division. They should be able to copy themselves. They should be able to generate lots and lots of tissue, not simply small amount of tissue. They should be able to specialize into different kinds of cell types in the body. And very, very important, number 4 bullet is very important. If they are transplanted in a human body for therapeutic purposes, they should not be rejected by the immune system of the body. They should be able to survive in the human body, not only survive, and live happily together with the neighbors. When I say live happily together, wherever they are transplanted, there are always other cells already residing there. So there should be a nice communication between the new cells which have been transplanted and the old cells which are already staying there. So this is what we mean by the word integration. They should be able to integrate nicely. They should, of course, perform their function for what they're being used in the body appropriately, and of course, not cause any harm in any way. So there is a huge, huge, huge demand for stem cell research. You can simply see by the numbers here how millions and millions of people are affected by all these diseases which are on the rise here, cardiovascular diseases, autoimmune diseases, diabetes is probably getting to be number one right now, and a bunch of other diseases. So many millions of people are suffering from so many diseases globally, and that's where stem cells can come into play and address some of the issues, not all the issues, some of the issues, depending on what is the major reason for these diseases. If it's genetic, they can be fixed through therapeutic cloning. But if there are complex diseases like cardiovascular diseases, the fix may be a bit more challenging. So there's a lot of potential for stem cell to address all these diseases here. All right, but we do know some people, even now, they are using embryos, not for therapeutic purposes, but actually for reproductive cloning, which is extremely unethical. There were a few reports that came out, I think, about 4 or 4.5 years ago from certain countries. And there are lots of ethical issues in using embryonic stem cells. Number one is, of course, you have to destroy human life, which is the donated embryo, in order to harvest the stem cell. And that's a big, big issue for several stakeholders. The second ethical issue is if someone has access to embryonic stem cells, they can actually start reproductive cloning and basically make what we call designer babies, where these babies are genetically engineered outside the body. And then once the engineering is done, these embryos are implanted into a woman's body, which of course is reproductive cloning. And thankfully that is prohibited. But this was done by a scientist 3 years ago, and he was jailed, and he's still in jail right now because he engaged in reproductive cloning and generated twins, female twins, to save them from getting AIDS, the disease AIDS. However, he actually did not follow any regulations, and that's how he actually landed up in jail. And then the third ethical issue is the use of induced pluripotent stem cell, which I'll talk about now, which is the most powerful tool in stem cell biology right now. As the name suggests, these are stem cells, these are pluripotent, but they are not embryonic in nature. Rather, they have been induced, they have been sort of like instructed to go back in time and revert back to their embryonic nature. So there's no embryos which are destroyed here, but I'm going to show you a little graph, a little picture, which shows you how induced pluripotent stem cells are actually derived from human stem cells. So other issues, we can go on and on talking about the issues, and anytime there's clinical trials, you want to make sure the patients are informed what their body tissues are being used for. They are equally protected, their rights and their dignity and their respect is very well maintained. And there's also ethical issues in animal research. So, you know, whenever scientists are engaged in research which is tied to human beings, there is a whole lot of regulation and oversight, and things don't get done quickly. That's why clinical trials take several years to get some drug into the market or a cell therapy into the market. So we can educate our scientists, our researchers and public, and make them aware how very important it is to engage in science which is good, which is beneficial, which is following all sorts of regulation and policies. And most important part, it is following all ethical guidelines. And the reason I have the slide here is because it is very easy to work with stem cells. And now we have tools which anyone can buy, and actually anyone can engineer a stem cell provided you have the right food. Because of the ease of manipulation of stem cells, there is a high potential of these stem cells to be misused, and like I said, could be used for reproductive cloning. So there is a lot of education which is available for both researchers and public to inform them about various guidelines which exist with regard to working with stem cells. So I'm going to end here very quickly here about I'm going to skip this slide, but this is what I wanted to show you. The induced pluripotent stem cell is actually a stem cell which is looks like an embryonic stem cell, but it's not coming from the embryo. In fact, it's coming from an adult cell, a very mature cell in your body, So let's say we start with your skin. You take a few cells from the skin and you add certain food into certain food in the growth medium of that cell, which is what we call genetic reprogramming. So essentially what we're doing in very simple layman languages, we grow skin cells in the lab. We put certain genes or certain food or certain proteins to allow them to grow. Now, what happens is, rather than going forward in time, they actually go back in time and they become more primordial, more original, and more embryonic-like. And that's why we use the word induced, because they have been induced to go back to an embryonic stem cell. And now what happens once you have iPSCs, now without having to destroy embryos, you can the iPSC cells and actually have them turn into any cell type in the body. So this was a major, major discovery by Dr. Yamanaka in Japan, for which he actually received the Nobel Prize. This was a monumental discovery that we could take a cell back in time to the early stages and turn it embryonic in nature. And now we don't have any ethical issues, because nobody's going to die by isolating a few skin cells from my skin. I'm not going to die. I just need to take a few skin cells, and lo and behold, they can turn into iPSC cells. So gone are the days where we used to be worried about killing embryos and isolating stem cells from that. That's all gone now. Most of the people are currently working with these induced pluripotent stem cells. And like I said, no need for embryos here. So these induced pluripotent cells have been turned into different cell types by various researchers. They have been turned into bone, cartilage, skin, neurons, muscle, lung cells, you name it. A lot of people have been successful into generating these very specialized cells from the induced pluripotent stem cells. So that's a great bargain here. And clinical application, they are very, very beneficial to work with. We can easily harvest them from body. We can have them regenerate or grow really fast. They can differentiate into different kinds. And we do not have to worry about rejection, because if I am harvesting my own skin cells and I'm turning them back into induced pluripotent stem cells and having them differentiate and put the cells back into my body, there is absolutely no question of rejection, because these cells are originally coming from me and it's going back into my body. So my immune system is not going to regard them as foreign, and they will survive in the recipient body, which is a huge, huge factor for success here. So despite iPSCs being used extensively for research here, there are certain caveats, particularly when we are using iPSCs for cell therapy purposes. There's a lot of science here, but I just want to mention to you that it's not very easy. And people ask us that, we've given $3 billion to the state of California for the past almost 20 years. Why haven't therapies come into the market? Well, there's a lot of things that need to be resolved here before we actually say, okay, here's a cure for Alzheimer's, here's a cure for Parkinson's. Because some of the food that we use for turning these iPSCs into various cell types is, some of this food, the genes, tend to have cancer-like features. And we want to make sure that we don't generate undue cancer in the body. So this is one of the reasons why, more research has to be done to be not able to use these cancer-causing genes for iPSCs manipulation. Secondly, for genetic engineering, we also use viral vectors. And knowing the whole COVID-19 scenario right now, anytime we use a viral vector, we want to make sure the virus actually does not cause any undue damage inside the body. It does not lead to disease. And then, of course, at the scientific level, we are still working out the protocol for changing these iPSCs to different cell types. So there's a lot of, I would say, exciting challenges, and lots is being done. But remember, from the point of the lab to actually coming into the market, we have to clear all these hurdles. We have to understand the function. We have to do clinical studies. We want to make sure it's safe. It's not causing any problem to the humans. We want to make sure there's no immune response. And then, of course, for the local biotech companies who are making these cell therapies, we want to make sure we understand who holds the intellectual property rights. And finally, the FDA has to give the approval, then only it'll come into the market. So this whole process may take anywhere from 9 to 16 years. All right, so I'll end this by telling you that the California Institute for Regenerative Medicine was created as a result of Prop 71. And this is where $3 billion was given. And this agency known as CIRM has given millions and millions of dollars to universities like us, where it has enabled us to train graduate students for the past 10 years and become skilled stem cell researchers. And there is now a huge demand for stem cell research in California. And of course, they've given a lot of money to the UCs as well as these big institutions to conduct clinical trials, which actually run into millions and millions of dollars. So CIRM is actually doing lots and lots of good work by providing us money to do both basic research and applied research in the area of biomedical sciences. So we are pretty thankful to CIRM for giving us funding. So there's lots of CIRM funded institutes, centers of excellence, special programs. There's lots going on in California today. And if anyone is interested in knowing more, one good source for understanding what's going on in the area of stem cell sciences is the International Society for Stem Cell Research, also known as ISSCR. And this is where you can find great amount of information of what's going on in the world today with regard to stem cell science. So I'm going to end here, and I thank you for your time and your attention. I will be happy to take questions later, but since it's already 10:45, maybe I would really request my students to get started now one by one, and we can take our questions towards the end of the talk. Okay, I'm looking at the questions right now, but I'll answer all your questions towards the end because my students have spent a lot of time, a lot of effort making these presentations, and I want to make sure they're given good quality time to present their work. So can we have the group of Martin Molnar and your colleagues please go first, and then we'll have the second group, Gaurav and his colleagues go second, and lastly is Mohammed and Maria and your colleagues will go in the end because I believe you have a little video you want to show towards the end, right? All right, so Martin, all yours.
So sorry, could we have screen sharing enabled so I can share the presentation?
I think Michael, have you given Martin the rights to share the screen?
Yeah, I can make that. Give me 1 second.
Okay. Yeah, if you can give it to Martin Molnar.
They should be able to share the screen now.
Okay, go ahead, Martin. You can share your screen.
Thank you.
And I want to remind all the 3 student groups, please keep your talk to only 20 minutes each, because we need time to answer the questions from the audience later. Thank you.
Can everybody see the presentation?
Yes.
Okay, good morning all. So today we want to talk about the potential of stem cells for treating diabetes. First of all, let's take a quick look on this disease. Let's see what diabetes is. Next slide, please. So diabetes is a chronic or long-lasting condition in which the body is not able to turn the sugar of the food that we eat or the drinks that we drink into the energy. The healthy bodies naturally produce a hormone called insulin, which enables the body to convert the sugar into energy, but in diabetic people, it's not gonna happen. So diabetes has 2 major types, type 1 and type 2. In type 1, the body is not able to produce the insulin. So that is why the patients need to take or inject insulin every day. In type 2, the body does produce insulin, but due to some special causes, the body is not able to use the potential of insulin to convert the sugar into energy. Both cases lead to accumulation of the sugar in the blood, which itself can damage different organs such as nerves, kidney, eyes, food. Diabetic people may also have some skin conditions, hearing impairments, as well as depression. Next, please. So in the right picture, you can see some causes of type 1. So in the middle of this picture, in the center, you can see an organ. It is the pancreas. It is the organ that releases insulin. So autoimmune disease, viral infections, autoimmune destruction of beta cells, for sure damage or removal of the pancreas, some special drugs and chemical toxins, unfavorable environmental factors, and endocrine disease, as well as hereditary predisposition can affect the potential of this organ, the pancreas, in releasing the insulin. So on the left, you can see a comparison between the healthy and diabetic. So in healthy people, the pancreas is able to release the insulin, which is shown in green dots here. Then the insulin moves the glucose to the cell. The glucose is the sugar of the food that we eat and are shown in blue dots here. So the insulin moves the glucose to the cells, but in diabetic patients, immune cells may destroy the cells, the beta cells of the pancreas, which are in charge of releasing the insulin. So the insulin is not released in this case. As you see, there is no green dots or insulin in this part. So the blue dots, which are the sugar, they just accumulate in the blood, and the other cells are not able to use them as a source of energy. Next, please. In type 2 diabetes, the pancreas is able to release the insulin. So these patients have sufficient amount of insulin. The insulin then goes into the blood vessels. But due to different reasons, such as obesity, some special genes, having a family history, eating or drinking much sugar, as well as having stress and high amount of stress hormones and lack of activity and exercise, the body, the different cells in body, like muscle cells, become resistant to the insulin. So they're not able to use the potential of insulin to convert the glucose into energy. Next, please. Here you can see some common symptoms of type 1 and type 2. On the left, you can see the type 1 symptoms. So patients usually first feel thirsty, they feel hungry, they have weakness, they may lose their weight, they may have a blurry vision. They have to go to the restroom a lot due to frequent urination. And if they cut a part of their body, like their finger, the cuts may heal really slow. In type 2, again, patients have frequent urination, constant hunger, slow healing of wounds, blurred vision. They may lose weight. And they may also have fungal infections, as well as numbness of hands and feet, especially in the fingers. Next, please. So it is interesting to mention that 1 in 10 Americans are diabetic, and 1.4 million of new cases are diagnosed each year. And 1 in 5 patients do not ever know that they're diabetic. And the highest amount of diabetes is found among Black and Latino patients, both in male and female. And diabetes is the 7th leading cause of death in the United States, and is estimated about $327 billion for the medical cost. And as it is shown in this picture on the right, you can see the number of diabetes patients are just increasing. Next, please. So adults with diagnosed diabetes may also have different conditions, may also suffer from different conditions. The majority of them will have a high blood pressure. They may have high cholesterol. They may have chronic kidney disease, and some of them will also have vision impairments and even blindness. Next, please. So here is the country distribution of diabetes. As you see in some states like Tennessee, more than 12% of the residents are diabetic. And in California, we can see about 10% of the residents are diabetic. Thank you. Next, please.
All right, thank you, Nigar. My name is Stephanie. I'm representing on type 1 diabetes stem cell therapies. But first, I want to go over insulin. I know most of you probably already know and are aware of insulin's uses with diabetic patients, but it's been used for the past 100 years, and it was a major breakthrough. It made the disease a much more manageable disease rather than a fatal diagnosis. Next slide. So basically how it works is in our pancreas, we have beta cells, which basically detect rising glucose levels in the blood. And when they detect this, they will secrete insulin, which will basically counteract the rising levels. The problem with type 1 diabetics, which Nigar already touched on, is that the beta cells are destroyed by their own immune system, so they do not have this self-regulating feature. So with the onset of insulin, it allowed patients to detect their own blood glucose levels and self-administer insulin. Next slide. So I just want to include this infographic. It's basically where we are currently and where we're headed as of 2022. We start out with insulin. We now have wearable devices, which people have like automatic insulin injections. But the future really seems to be grounded in stem cell therapies and regenerative medicine. Mostly beta cell implantation and replacement therapies. Next slide. So one such therapy, it's a really exciting new medicine. It's by the company ViaCyte. So they have 3 stem cell therapies in their company's pipeline as of 2022. And these are undergoing clinical trials currently, but it basically involves medical devices that consist of pouches with beta cells inside these pouches, and they get implanted into the patient's bodies. Next slide. So here's the pouch. You can see the beta cells are on the inside, and on the outside of the pouch, the blood vessels of the patient are intended to grow around the exterior. And this design is basically to allow for a constant transfer of essential nutrients and proteins like glucose, insulin, oxygen, basically to allow those cells to grow at optimal conditions to successfully replace the destroyed cells. And the pouch is also very interesting because it allows for protection from these beta cells from the immune cells of the patient. And this is very important to prevent an immunosuppressive attack, which can basically destroy all the implanted cells. Next slide. And here's just a realistic picture of the pouch. As you can see, the cells are on the inside, the blood vessels are growing on the outside. And yeah, once again, these are in the clinical trial phase at the moment. I think they're still in the preliminary phase, but so far the company has reported success. And it'll be interesting to see in the next couple of years where this therapy goes, because this company is saying it could be a cure for type 1 diabetics and maybe even some type 2 diabetics. I'm now going to pass on to Patricia. She's going to cover type 2 diabetes. Thank you.
Thank you, Stephanie. So I'm going to be covering type 2 diabetes. So before patients are diagnosed with type 2 diabetes, some will actually be told that they are pre-diabetic. So during the pre-diabetic phase, there's the blood sugar is not as high as in the diabetic patients, but still not considered normal. So then the patients will have no symptoms, and it's also a reversible, it's reversible because patients could actually exercise and maintain a healthy diet That way their blood sugar levels will go back to normal. So for patients who are already diabetic, usually they need to have a constant diet, exercise, take medication daily, and also sometimes take insulin therapy. Next slide. So the pancreas, it usually measures the amount of glucose in the blood. So after a person has a meal, the blood sugar rises, and this actually causes the pancreas to release the peptide hormone insulin, and this causes the cells in the liver, muscle, and fat tissue to take up the glucose from the blood and stabilize the blood sugar throughout the body. As in, you could see type 2 diabetes, the insulin does not stimulate the cells, and this actually does not allow the other cells to take up the glucose. Next slide. Okay, so some medications that type 2 diabetic patients take are Nesina or Arseni, which stimulate pancreas to produce insulin. Another type they could take is Avendia, which inhibits production and release of glucose from the liver. Gazano and glucovins also improve insulin sensitivity in cells. Farxiga and Jardians inhibit reabsorption of glucose in the kidneys, and other medications break down starchy food, which include glycine. Next slide. Okay, so the beta cells are the ones that take up the insulin. They are found in the pancreas in clusters that are known as islets. So as mentioned before, the type 2 need to work harder to produce and release this insulin. And this is where stem cells come into place. Next slide. So stem cells are actually retrieved from the patient. They are then enriched in the lab and reprogrammed so that they could become pluripotent. They are then differentiated into beta cells using certain nutrients. And then the beta cells are transplanted into diabetic patients. So far, this currently is going on in animal studies, and it has shown that it provides prolonged insulin dependency and metabolic control, as well as prevents severe hypoglycemia. So more research does have to be made, especially in humans, to understand if the beta cells actually are helping diabetic patients. Thank you. I'm going to pass it on to Martin now.
Thank you. So in this segment, I'm going to go over the advantages of stem cell therapies compared to traditional treatment methods and analyze how exactly they improve patient outcomes. First of all, in type 1 diabetes, they offer the possibility, and it's important to note that it's just a possibility of a cure. So this means not just treatment of the symptoms, this means complete remission and insulin independence. So no daily injections, no pumps or other devices. Once the treatment is successful and produces enough insulin, treatment is complete. With injections, there is also the possibility of hypoglycemia, which is not an issue with stem cells. However, there is a risk of immune rejection and teratoma formation with stem cells. Teratoma is a type of tumor, and this is an inherent risk of any treatment involving stem cells. Another treatment option that's currently available for type 1 diabetes is islet cell transplants, as mentioned by Stephanie. So this requires the donation of a pancreas, which means that someone with the right profile has to pass away for the patient to receive a donor organ. And they take islets from the pancreas and transfer them into the liver of the patient. And this is successful about 60 to 70% of the time. And those patients achieve complete remission. after 5 years. However, it is not easy to isolate the beta cells from the donor organ, and most people actually need 2 transplants. And as you might imagine, there is a very significant shortage of pancreas donors. So with stem cell treatment, you don't need a donor. You are your own donor. Additionally, there is research being done on treating diabetes-associated complications. So diabetic retinopathy, neuropathy, and neuropathy are all conditions that could be treated with stem cells, but are not able to be treated with existing methods. Type 2 diabetes, there is a wide range of outcomes, as you saw earlier, and pre-diabetes is reversible, and even fully developed type 2 can go into remission, unlike with type 1. There is also a wide range of medications available, so stem cells might not be the right treatment for everyone. And it has been found that administering stem cell therapies to type 2 patients, non-obese patients and patients who have had the disease for less than 10 years are the ones with the best outcomes. So even if a stem cell therapy is administered, making life changes is still an important and necessary step in treating type 2 diabetes. So these are the challenges currently that have to be overcome before stem cell treatment can reach its full potential. The number of beta cells or beta-like cells that are generated in the Petri dish before implantation has to have a higher efficiency, a higher yield. And this means that inducing the stem cells into becoming insulin-producing cells has also been increased. There's also more work that remains to be done on protecting cells from autoimmune attack. So why is this necessary? Because as has been mentioned many times before, one of the big advantages of stem cells is that they are not going to be attacked by your immune system because they are your own stem cells. I mean, your own cells. The reason this is a problem with type 1 diabetes is because it's an autoimmune disease. So even though they are beta cells from your own body, that's the whole disease pathway of type 1 diabetes. Your immune system is going to attack your own beta cells. So they have to be protected in some way. And this can be achieved in biological ways, but also, as you saw with ViaCyte, it can be also achieved by physical separation. And finally, the last milestone is to enable complete insulin dependence, no partial, complete insulin independence. So not just partial independence, no repeat treatment, just complete independence with after the treatment is done. Okay, finally, there is a word of caution. There are already many companies that advertise and offer stem cell treatments or gene therapies in the US, but also abroad. And while stem cells have enormous potential and there are hundreds of treatments in different stages of clinical trials, these are generally not available to the public yet. The vast majority of adverts right now are scams. The FTC has warned about them, the FDA has warned about them, and I included links for people who are interested. Some people travel abroad to get treatment from less regulated countries and then come home with a tumor. We have to remain patient and await for FDA approval. There is currently only 23 approved stem cell products, and I included a list for that too, if anyone is interested. Thank you for your attention. And as a goodbye, I included a picture of a beta cell, and be happy to answer any questions.
Thank you so much, Martin, Nigar, Stephanie, and Patricia. That was really nice. Thank you. So I think we'll take the questions towards the end because I want to make sure the other two groups have enough time for their presentations. So we'll hold off on the questions. And can we have Gaurav Saini and his group please launch your presentation? And Michael, if you can give the share in rights to Gaurav. Oh, okay, he has it. Go ahead, all yours. Thank you.
Good morning, everyone. So our group is presenting on the Alzheimer's disease and the stem cell research related to it. And Rayan will begin the presentation with the introduction of the Alzheimer's disease. Rayan, you can take over.
Hi, Ryan. I'll be starting off my group's presentation on Alzheimer's disease and stem cell research. What is Alzheimer's disease? Well, Alzheimer's disease is a neurodegenerative disease. It is characterized as impairment of behavioral and cognitive functions, including memory, comprehension, language, reasoning, and judgment. Alzheimer's is a type of dementia and accounts for 60 to 80% of dementia cases. I'll be listing a couple of common signs and symptoms. One of them is confusion with time and location, difficulty completing familiar tasks and solving problems, memory loss, misplacing items, poor judgment, and unfounded emotions. Here is an image of disease progression. On the bottom left-hand corner, you can see that this is an image of a healthy brain without amyloid plaques, which is the cause of Alzheimer's. And as you see in the middle image, it shows the progression of Alzheimer's, like in the beginning. And by the right-hand side, you will see the full severity of Alzheimer's disease. Here is the statistics of Alzheimer's. As you can see in the orange right about here, people who are 85 and over have there's been an increased number of people who have been diagnosed with Alzheimer's, and the same could be seen for people between 75 and 84 and age group of 65 to 74. It has been steadily increasing, but it's not as bad as age groups of 85 and up. And here on the right-hand side is projected rise in Alzheimer's cases by 2025. As you can see here in California in general, there is a possibility of a 26.5% to a 34.8% possible increase of Alzheimer's. And right now I'll be passing off to my peer and friend, Charlotte.
Hey, hi, I'm Charlotte, and I'll take you through the worldwide distribution and some risk factors associated with Alzheimer's. So Alzheimer's affects people globally, but the question is to what extent? Worldwide, there are about 46.8 people living with Alzheimer's and any other forms of dementia. And this distribution seems to vary with cultural differences among the nations and to a larger extent, continents. You can see here that the most affected continent is Asia, followed by Europe, the Americas, and Africa. Next slide, please. Okay, so your risk for Alzheimer's disease goes up as you get older. For most people, this is age 65 and upwards. And also your risk of developing the disease is also higher if you have a first degree relative, say your parent or your sibling getting the disease, or you inherited the disease from your parents' genes. And also it's been known that people with MCI, that is, they also have a significant risk of developing the disease as the disease progresses to developing dementia. And also unhealthy lifestyles like smoking, excessive alcohol intake, poor sleep patterns, and bad heart health could also be linked to the disease. And people with Down syndrome also develop the disease later in life. Next slide, please. So currently there's no cure for Alzheimer's disease. However, the FDA has approved 2 types of medications. That's the cholinesterase inhibitors and the memantine. And these drug classes manage the disease by temporarily easing the symptoms and also slowing down disease progression to improve quality of life. There are other therapies like monoclonal antibodies and stem cells that are currently under clinical investigation as they could be a potential cure to the disease. Next slide, please. So the disease typically progresses in 4 different stages. We have the early stage, followed by the mild, moderate stage, and finally the advanced or severe stage. Most medications will work best at the early stage as they will slow down disease progression. And during the mild or moderate stages of the disease, you normally need counseling and support groups to guide you on how to manage the changes, because at those stages you see some little changes in the disease. So you need those counseling to help you manage the changes that come with the disease. And this is done through psychosocial, behavioral therapies, and also through physical therapies. And at the advanced stages of the disease, where the person is unable to communicate well or look after themselves, there's a need for complete caregiver support that helps them with their daily tasks. Next slide, please. All right, so I'll hand over to my colleague Gaurav, who will take you through the ways to prevent Alzheimer's and also talk a little bit about how it's diagnosed.
So I will be discussing some ways by how we can prevent the Alzheimer's disease. So these are some points. So every one of us should exercise daily, even if we are not at high risk of getting Alzheimer's. A brisk walk for about 15 to 20 minutes can be helpful for improving the blood flow to the vital organs. So we should prefer the Mediterranean diet. Eating the Mediterranean diet is crucial because it has green vegetables, legumes, and it should also include some good and healthy fats. We should kick out all the bad or the unnecessary habits like smoking, and we should also reduce our drinking to only one drink per day. It should not be beyond that. And although above these points help you in maintaining the good health, but if you are already suffering from any disease like high cholesterol or diabetes, taking the regular medications can protect your vital organs like brain in such a case. So the next step is like we should stay safe. We should stay safe from the accidental falls or the mini strokes or serious illnesses like brain infections like meningococcal infections through the proper vaccination, which can hasten the disease. And we should also engage ourselves in the brain challenging activities, or we should also participate in the volunteering activities or join a club with the friends that can help to release the happy hormones like the dopamine, which can prevent the Alzheimer's disease. Next slide, please. So this slide talks about the various exams or the that can help in the diagnosis of the Alzheimer's disease. So the first one is the complete and the accurate medical history. A thorough medical history is taken for any suspected patient of the Alzheimer's. It can be very likely that if there is Alzheimer's in the family history, it can be found in the future generations, although it is not a guarantee because many epigenetic changes also occur, which are beyond the genetic prediction. So the next test could be the mental status test. In this test, the various mental ability tests like recalling a place or any event or any problem solving test can be done. The next test is full physical exam test or full neurological exam test that is conducted by the physician. The new techniques include the brain imaging techniques and the blood tests. The blood test like plasma PTAU 181 can be done because in the patients of the Alzheimer's, there is a protein called as TAU 181 protein, which is raised in the plasma level of the patients. And the brain imaging tests like PET and the MRI can also be done. PET is the positron emission tomography test, and MRI is the magnetic resonant imaging test that can be done for the Alzheimer's. Next slide, please. I think you skipped the slide, Rehan. Can you? Yeah, this one is the slide. So these are the various types of tests that can help in the detection of the Alzheimer's in the early stages. So it was very difficult to predict or make a diagnosis for Alzheimer's without the advanced technology. But it can be seen from this diagram that with modern sophisticated techniques, we can predict the occurrence of the Alzheimer's or the likeliness of its happening in very early stages. For example, the CSF tests can help detecting the Alzheimer's as early as 20 years, and the brain imaging techniques like PET and MRI, they can help in detection of the Alzheimer's or making a definitive prediction about the Alzheimer's up to 15 years in advance. And the memory tests can help in detection of the Alzheimer's up to 10 years before the onset of symptoms. Although we have all these methods, but final diagnosis of the Alzheimer's can only be made after the final onset of the symptoms. Next slide, please. So all of these slides do raise one question, that why the current treatments are not effective for the Alzheimer's. And it can be seen from this diagram that majority of the drugs, they fail to develop a successful therapy for the Alzheimer's. And most of the drugs, they fail after the phase 1 trial. Like as you can see in the diagram, like 72% of the drugs, they fail in the phase 1 and they don't go to the phase 2. And only 8% of the drugs goes to the phase 2. And when until you reach the phase 3, only 2% of the drugs are successful of going to the next stage. So a decade's worth of clinical trials identified only one approved drug, and 99% of the 413 trials, they failed to produce the drugs. And total of about 244 compounds were tested from 2002 to 2012. So the main reason why this treatment is not effective is because it is very, very hard to find the cure, because the symptoms of the disease or the confirmation diagnosis is made only when the irreversible damage to the brain cells has occurred. And another reason is that it is difficult to find people for the conducting clinical trials when the patients are in the initial stage, which makes it hard to develop successful medicine or drug for the Alzheimer's. So another reason is also that Alzheimer's, it can affect different people very differently. So one drug which might work in one person may not work for the other person. So stem cell therapy can be a boon for the Alzheimer's patient because it works on the principle of regenerative potential of the cells. And my colleague Robin will be discussing the stem cells, what they are, and what are the possible therapies that can be associated with the stem cells for the Alzheimer's disease. Next slide, please. So Robin will take over from here.
Yep. Hi, everyone. My name is Rovin, and today I'll be presenting a deep dive onto the topic of stem cells and more specifically their influence on Alzheimer's treatment. Now, first, what are stem cells? Stem cells are the body's raw material. They are cells from which all other cells with specialized functions are generated. And under the right conditions in the body or in a laboratory, stem cells divide to form more cells, also known as daughter cells. These daughter cells become either new stem cells or specialized cells, also known as differentiated cells, that also have more specific functions. These include blood cells, brain cells, heart muscle cells, bone cells, etc. This feature is significant as no other cell in the body has the natural ability to generate new cell types. Next slide, please. Now let us discuss one of the types of stem cells researchers are currently utilizing. Induced pluripotent stem cells are derived from the skin or blood cells that have been reprogrammed back into an embryonic-like state that enables the development of an unlimited source of any type of human cell that is needed for therapeutic purposes. These cells hold great promise in regenerative medicine as they can increase indefinitely and give rise to other cell types in the body, such as neurons, heart, pancreatic, and liver cells. Thus, they represent a single source of cells that could be used to replace any of those lost due to damage from disease such as deterioration caused by Alzheimer's disease. Next slide, please. How stem cells can benefit you. You may ask yourself, why is there such interest in stem cells? Well, researchers hope to use stem cells to increase their understanding of how diseases occur and generate healthy cells to replace cells affected by disease through a concept known as regenerative medicine. Stem cells have also shown potential to be grown and become new tissues for use in transplant and for regenerative medicine. And in addition, we can use stem cells to test new drugs for safety and efficacy before using investigational new drugs on humans that could potentially have harmful side effects on us. As such, a concept that we'll delve into in a little bit is a newer area of study involving human stem cells that have been reprogrammed into tissue-specific cells to test new drugs. Next slide, please. Now let us discuss the cells that are specific to the brain, because we're talking about Alzheimer's research here. Neural stem cells are largely undifferentiated cells originating from the central nervous system. They have the potential to give rise to daughter cells that grow and differentiate into neurons and glial cells, which are found in the brain. Neural stem cells are active in almost all areas of the brain and were first found to be involved in the brain's hippocampus, which is the area of the brain that is instrumental in the formation of memories. As you can see from the diagram, there are a couple of different sources using recent technological advances that we can derive these cells from and eventually make neural stem cells. These include direct isolation and reprogramming of a person's tissue, such as their skin cells. They can also directly harvest cells from a recently deceased person's neural tissues, and reprogramming of embryonic stem cells, which are stem cells that are taken from early stage embryos. Researchers believe we can use neural stem cells to regenerate cell deterioration caused by neurological disorders such as Alzheimer's. Next slide, please. I added this diagram to illustrate the mechanisms and actions that researchers have used to use stem cells to treat Alzheimer's disease. We can dive into specific examples in a moment, but I wanted to show you just the broad scope of stem cell treatments. These include providing anti-inflammatory effects, homing to damaged tissues, recruiting other cells to aid in regeneration, supporting tissue remodeling over scar formation, inhibiting cell death, and differentiating into necessary tissue to regain the brain's functions. Next slide, please. Stem cell biology is rapidly advancing in the field of research and has been contributing to a substantially diverse array of scientific disciplines, ranging from developmental biology to, of course, regenerative medicine. In recent years, drug discovery has been one of the most promising applications for stem cell biology, where stem cells are increasingly being used in new and innovative ways to improve the drug discovery process. This can apply to academia, biotech startups, large pharmaceutical companies, etc. In the case of using stem cells specifically for treating those with Alzheimer's disease, researchers can use disease modeling where they can genetically reprogram normal differentiated adult cells back into an embryonic-like state, iPS cells, and then these cells can then be transformed into brain cells and cultured in a lab that will simulate typical neuronal activity found in the brains of those with Alzheimer's. Doing so can allow researchers to model the typical brain behavior of an individual with Alzheimer's, and we can use this information to test and produce drugs that can relieve the effects of Alzheimer's disease and hopefully one day discover a cure. Next slide, please. Now, I just wanted to end today's presentation on Alzheimer's disease treatment using stem cells with a brief look into current and future research on stem cells. Here, researchers examine the use of one potential application of stem cells known as cell replacement therapy. This is where human iPS cells are derived from a patient's own somatic cells and serve as a cell source for clinical transplantation therapy. One essential feature of this research is to use genetic repair to develop iPS cells that have regeneration from deterioration specifically caused from Alzheimer's disease. And ultimately, these stem cells will allow us to continuously culture and transplant healthy neural cells into humans' brains, and hopefully one day serve as a cure for Alzheimer's altogether. This ends our presentation for today. Thanks for watching.
Thanks so much, Gaurav and crew, for taking us on a journey to Alzheimer's disease. And we will have the third group, Mohammed and Maria, and I believe two others, I'm so sorry. But please go ahead and project your presentation.
Hello everyone, this is our group. We are talking about today about the spinal cord injury and stem cell therapy. In our group, I am A. from Nazmulak Santo, Mohammad David, and Maria Carmen Arianes. Next slide, please. We know everyone, the Superman actor, the Christopher Reeve. He's got accident and spinal cord injury, and he is a very good officer of stem cell research on the spinal cord injury. He is the actor who showed the world the meaning of courage after the spinal cord injury, and he supported the stem cell research and helped it to emerge as a major campaign issue between the President Bush and Senator Kerry. Next slide, please. Yeah, my friend Carmen will present this.
So the function of the spinal cord is to communicate between the brain and the body. So a spinal cord injury is the loss of communication between both the brain and the body. There's two levels of spinal cord injuries. There's the complete spinal cord injury, which is permanent damage to the spinal cord. This will cause paraplegia, which is complete paralysis, and tetraplegia, which is inability to move the upper and lower parts of the body. And there's also incomplete spinal cord injury, which is partial damage to the spinal cord. And patients still have the ability to move and feel depending on the spinal cord injury. Next slide, please. So the spinal cord is surrounded by rings of the backbone called vertebrata. These bones constitute the spinal cord column or the backbone. The higher the spinal cord injury on the spinal cord column, the more dysfunction a person will experience. So there's 4 areas where spinal cord injuries can happen. There's the first one right here is the cerebral spinal cord injuries, which affect the head and the neck. And then we have the thoracic spinal cord injuries, which affect the chest, the mid-back, and abdominal muscles. And then we have the lumbar spinal cord injuries, which affect hips and legs. And then we have the sacral spinal cord injuries, which affect the hips, the buttocks, and the back tights. So right here on the right side, you could see like a diagram of this areas of the spinal cord that get affected by them. Next slide, please. So the cause of spinal cord injury is the physiology of the spinal cord is damaged. So there's cellular interactions among the spinal cord that are interrupted or damaged from external trauma. Usually this trauma is caused by car accidents, which is the most common cause, falls. And we also have other external traumas like violence, sports, and medical surgeries. But this is spinal cord injuries could also be caused by non-trauma, such as cancer, tumors in the spinal cord, inflammation, or like blood loss to the spinal cord. There's also multiple sclerosis that could cause it, such as osteoporosis. Next slide, please. So statistically, there is 1,298 cases per 1 million people globally of spinal cord injuries. And here in the United States, we have approximately 294,000 cases of spinal cord injuries. Basically, 54 cases per 1 million people of here in the United States that have spinal cord injuries. So this actually affects more males. So there is 78% of spinal cord injuries are males, and the age in the 1960s, the demographic for people that had spinal cord injuries was 29 years of age. And in 2015, that actually changed to 43 years of age. So this is actually a very expensive injury. It's expected to be more than $3 million of cost for people that get spinal cord injuries. And it has a high mortality rate, especially in the first year of getting a spinal cord injury, where people actually get sick of respiratory, circulatory, or cardiovascular problems from this. Next slide, please.
Yeah, I have the stem cells. Stem cells are the body raw material, cells from which all other cells' specialized functions are generated. So under the right conditions in the body or a laboratory, stem cells divide to form more cells, which are called the daughter cells. Next slide, please. So for the stem cell therapy, it is a treatment that uses the stem cells or cells that come from the stem cells to replace or to repair a patient's cells or tissues that are damaged. The stem cells might be put into the blood or transported into the damaged tissue directly, or even recruited from the patient's own tissues for cell repair. Next slide, please. So there are the three main kind of stem cells: embryonic stem cells, adoptive cells, and cord blood stem cells. Dr. Parmar already discussed about this, so I not longer this the slide. The next slide, please.
Hi, hello.
Yeah.
My name is Mohammed. I am pleased to be with you here today, and I would like to thank Dr. Parmar for such an opportunity. So we're going to discuss the pathophysiology of this injury. So basically, the spinal cord injury is composed of interrelated events. Each serves as a facilitator for another. So therefore, we have two main phases, like the primary injury, like right after the accident, or any cause that could cause the fracture or any mechanical stress on the spinal cord. And this and also there is a post primary injury, which is the secondary injury, which is composed of three main subphases. And the first phase is the acute phase, which range between the time of the injury till like 2 days after. And there is the subacute phase, which is after 2 weeks, and the chronic phase, which is the most, I would say, dangerous phase, which causes paralysis and loss of control of your limbs. That could range from days to years. And there is like a MRI pictures comparing the normal spinal cord versus injuries over the phases. As we see here in the pointed through the red arrow, the area of the lesion, as the time goes, the injury gets aggravated, and we see like formation of a certain object through the subacute phase and going to the chronic phase. And this object is called the glial scars, which will affect the blood flow, the transmitters of neuron messages. Basically, it affects the communication from the brain till like the limbs, the legs, and any functional movement of the body. And also there is some medical approach, and unfortunately the medical approach is targeting the pain relief and the pain management. So for example, we listed like three, like the most popular drugs taken for this condition. We have methylprednisolone, which is a very strong anti-inflammatory, naproxen, which is a pain reliever, and gabapentin, which is like a pain reliever, especially for nerve pain. And also there is non-pharmacological approaches. So most patients who suffers from the spinal cord injury, they're treated via massage therapy to improve the upper body muscle strength and range of motion and enhance function. And also acupuncture comes to use here for and also it's widely used for improving the recovery of the motor sensory function in patient with spinal cord injury. And also there is an electrotherapy, which is the transcutaneous electrical nerve stimulation, which is mainly used as a pain reliever. And also the stem cell therapy for the SEI. So there is like 4 main sources of the stem cells. They could come from the bone marrow, umbilical cord, adipose tissue, which is like the fat cells, and amniotic fetal stem cells. And these stem cells could improve the communication between the neighboring neurons and the further neurons as well. So it increasing the signaling, especially the chemical signaling, and also the transplantation of stem cells can promote axonal regrowth, and can also modulate the neuroinflammation, and also has an effect on reducing the glial scar shown in the MRI pictures. And not only that, but also it supports remyelination, which is the protective layer, like the coating layer of the neuron. And one of the effect of the spinal cord injury is the degradation of that layer. And there is 3 main ways of stem cell administration. So we have the intravenous, which is easy to use and doesn't require like special equipment. Excuse me. However, the number of stem cells derived to the spot of the lesion is very small. And then another way of administration is the enterical, and can deliver a large number of cells to the spinal cord using the spinal fluid. And the last way of administration, it targets specifically the spot of the lesion, and requires a very delicate surgical intervention. And here is like a list of most used stem cells. For example, the bone marrow stem cells, it promotes axonal excuse me, regeneration, reduce glial scarring density, regulate autophagy, elevate neuropathic pain, and also the embryonic stem cells promote osteolysis and support nodal architecture, and also attenuate pain. And the induced pluripotent stem cells promote axonal sprouting, inhibit demyelination, promote synapses formation, means like the communication between the neurons, and also inhibit glial scar, reduce lesion size, and improve the respiratory function, which could be affected by the injury, especially if it's like right in the neck, which is affect the respiratory system. And also here we're pleased to present some successful stories. As we see in this man is able like to stand straight using his limbs. He has been sitting in a wheelchair for a while, and this patient suffered from severe spinal cord injury and received intrathecal injections of autologous adipose-derived stem cells. Basically, they used the fat cells from his own tissue. So your medicine is in your own body. And as we see in the picture, the patient exhibited significant improvements in the function of his upper and lower extremities. And the last slide, I leave you with Anthony. With his feedback, Anthony received the allogenic umbilical cord stem cell transplantation, and there is his feedback here.
My name is Anthony Paulette. I'm from Puna, Hawaii. I live in Humboldt County.
What happened?
My name is Anthony Paulette. I'm from Puna, Hawaii. I live in Humboldt County, California. I was in a car accident, a very bad car accident. I had 7 fractures and three breaks. It was my pelvic bone got broken 4 spots. My sacrum fractured. My urethra got cut. I had 3 broken ribs, a fractured arm, and a crushed hand. It was, I think, 50-50 chance of living. They told me that it was going to be really hard for me to walk again, that I was going to have to probably teach myself to learn how to walk again. It'd probably be quite some time. The first time I came here, I was in a wheelchair. I just got my pelvic. They said my bones were stable a little bit, so my fractures were stable finally. It was like 6 weeks after my accident, and I came here. I got the stem cell, and I'm walking now. It's like 2 weeks later. I was walking before I left here the last time. I feel like I'm having a second chance at life because of this, stem cell treatment. I mean, I can totally walk now. It's amazing, without a limp or anything. And without Dr. Leslie's help, I don't think I would be nearly where I'm at. About a year after my accident. I can walk on my hands now.
And thank you so much for your time. And this is the end of our presentation.
Thank you so much, everyone, all 11 of you, for these wonderful presentations on such varying, diverse issues like diabetes, which is a metabolic disorder, versus Alzheimer's, which is a neuronal disorder, as well as spinal cord injury, which is sort of not a natural disaster, mostly through injuries and accidents. So thank you so much. And we will open up the session for questions. If the audience has any questions, please go ahead and pose your question, either on the chat or you can actually speak out your question. Anybody has any questions? We are happy to answer your questions, either myself or any of my students. Okay, since we don't have any questions, I would like all 11 of my students to let all of you know these are all stem cell students, which means they will be starting their one-year internship very shortly in the month of September. And they will be actually finishing up the internship next year in August. So for 12 months, they will be off campus and conducting the stem cell internship. So can each one of you please undo your videos and quickly give like a 10-second information about where you'll be interning, whose lab, which institution, and basically broadly what area of research you'll be interning, so that a wonderful audience gets to know how we are advancing the cause of stem cell research. Please, I may call upon you based on the names I'm seeing on my screen. We'll start with Stephanie.
Hi, I'm Stephanie. I'm going to be interning at Cedars-Sinai in Dr. Arun Sharma's lab, and he does cardiovascular research with stem cells.
Thank you. Nigar? You muted, Nigar.
Thank you. So, hi, I'm Nigar. I'm going to be an intern at USC, Dr. Giorgio Quadrado's lab. I'm going to work on neurodegenerative disease, and our focus is going to be on autism. Thank you.
Patricia?
Hi, my name is Patricia, and I'm going to be working at Cedars-Sinai with Dr. Shane Dementri, and we're going to be working on the skeletal muscle and on stem cells. Thank you.
Martin?
Hi, I'm Martin. I'm going to be doing research at UCSD under Dr. Miles Wilkinson, and we are doing research on spermatogonial stem cell creation and curing impotence.
Gaurav?
Hello, everyone. I'll be based at the UC Davis in the Northern California, and I will be working under Dr. Kit Lam, and our project would be based on by using these stem cells for different types of cancers like ovarian cancer, breast cancer, and oral cancer. And thank you. Thank you, everyone.
Robin?
Yeah, everyone. I'll be studying at at Stanford University. I'll be working in Dr. Charles Chan's lab, and we'll be doing cancer immunotherapy, specifically for blood cancers and bone regeneration.
Great. Charlotte?
Hi, everyone. I'm Charlotte, and I'll be interning with Dr. Gleason at the UC San Diego, and we're going to be, our work is going to be on brain cancer cells using neural stem cells. Thank you.
Ryan?
I'll be also be working at UC San Diego, also in Dr. Joseph Gleason's lab, and I will also be working on brain cells, particularly in a neurodegenerative manner, like autism.
Shayanto?
Hello everyone, I'm Santa. I'm going to the Beckman Research Institute, City of Hope, in the Young-Hong-Si lab. My focus on the disease modeling and drug discovery of neurodevelopmental disease. Thank you.
Mohammed?
Hi, I'll be working with Dr. Talbot at UC Riverside. Our main focus will be the effect of tobacco products on the stem cell development, especially the flavoring chemicals.
Especially in the e-cigarettes and the vaping devices.
Yeah.
And finally, but not the least, Maria.
Hi, my name is Maria, and I'm going to be working with Dr. Crystal McCall at Stanford University. We're going to be doing gene therapy to treat cancer. She does, her main focus is CAR-T gene therapy research.
Thank you so much. So I just want to let the audience know that we have fantastic students on our campus. And you can see by the very diverse nature of their research, they are working in all areas. I mean, you name it, cancer versus e-cigarettes and development of the fetus, autism, neuronal disorders, and metabolic disorders. And we actually place our interns all over California. Pretty much all the UCs. We also place them in USC, Stanford University, Children's Hospital of LA, Cedars-Sinai, City of Hope Beckman Center, Scripps and Salk institutes. So you can see we are placing our students in the best of the best institutions, not only in the US, but these are the best in the world. So we're very proud of our students, I am particularly, and very thankful to CERM for giving us the funding for 5 years, which allows us to fund these students. Funding is much needed for all of us, particularly with the COVID challenges. So really thankful to CERM to provide them both scholarship as well as living expenses funding, and to enable them to give talks like this to a wonderful audience like you. And thank you for bearing with us for exactly 2 hours now. But I do see just a few questions here. I'm quickly going to answer.
I have a question.
Yes, please go ahead.
Where are the embryonic stem cells stored, and what happens to them if they're not used for this research?
Good question. So the embryos are normally stored in freezers at fertility clinics. Mm-hmm. And typically when in vitro fertilization is done, when a couple is seeking IVF procedures, normally they have about 10 to 12 embryos, and couples normally don't implant more than 2 or 3. So the remaining 8 or 9 are lying frozen. And once the couple signs off and says they don't need any more children, they're eventually destroyed because the freezers can only hold so many embryos in tiny vials. And we're talking about, hundreds and thousands of people who are going for IVF. So they are frozen unless they are passing through the policies of being harvested for research. Otherwise, they are actually destroyed.
Thank you.
I have a question for you regarding the research that these, and first of all, thank you for this amazing presentation by everybody. The students that are now going on to do research, and I assume they're going to be using the adult stem cells that you talked about and, converting them to the iPS stem cells. Where will those original stem cells come from? Do they purchase it? Do they use people who come in and donate those, or where will they get those to conduct the research?
That is an excellent question. So there's 2 ways by which they can actually do work with iPSCs. One, the iPSCs are available through the ATCC, that's the American Tissue Type Collection. And you can order an iPSC line for about, I think it's about $400 or $500 or even longer, but you need to order it only one time. Then you can grow it and make stocks and freeze it. Now, in other cases, when the students have to study diseases, they cannot do with normal iPSCs. So in those cases, they actually get skin cells, particularly fibroblasts, from actual patients. And the students don't get to see the patients because the labs where they're going to actually intern, they have a collaboration with various hospitals across California, from where the patient stem cells and the fibroblasts are ordered, or not ordered actually, you don't pay for them, but they are supplied from there. And then in the lab, they are turned back into time to iPSC. So there's two ways. One, ready-made order for normal iPSCs. And if they are patient-derived iPSCs, then it goes through the whole protocol of institutional review board, because now you're dealing with human tissues. So both ways. And
thank you very much.
A lot of our students actually, the first 3 months of their internship is going to be focused on how to create iPSCs from typically fibroblastic cell lines. That's what most of them, I would say most of them would be doing. So they'll be pretty well trained by the end of the year. Any other questions? I'm just going to look at a few other questions here. So there is a question here. I'm just going to go down the list really quick. So there's a question from Ralph. Is the progress rapid and how is the funding? The progress is, I would not say rapid, because we have to be very wary of the safety issues. And particularly when Martin actually said stem cell transplantation can actually lead to the formation of teratomas, which are basically neoplasms, tumors inside the body. And we don't want that to happen under any condition. And the reason why teratomas do arise is solely because of the feature that I talked about of the stem cell. Their self-renewing capacity can be sort of like a bad blessing in disguise, so to speak. Because of this very feature, they can actually lead to formation of tumors. So I think all these safety issues, regulatory issues, so the progress is there, but I would not put it as rapid, but it's much faster than it was, let's say, 19 years ago. It's much, much faster. In terms of funding, there is plenty of funding in California through the California Institute for Regenerative Medicine. And now with the discovery of iPSCs, the induced pluripotent stem cells, there's a lot of funding even coming from the National Institutes for Health, the NSF, as well as private donors. So there's a lot of funding. And particularly in San Diego is the real hub of stem cell research, and Stanford University, two areas where there's a lot of private donation being made for creating these buildings of stem cell research. And they are tapping into the best minds from the world to come and work there on disease-focused areas where stem cells can benefit. Second question by Bruce is, how do the candidates get selected for the therapies? Now, that's an excellent question. I think that's a pretty rigorous protocol where the candidates have to exhibit certain levels of disease progression, age, as well as symptoms, and also subject to the fact that they have not undergone a previous therapy, which could actually cloud the impact of actual stem cell therapy. So I would say lots of criteria for selection, because I'm sure a lot of patients want to be in a clinical trial, but not everyone can be, particularly for phase 1, where only about 30 or 50 are chosen. And then phase 2 is about 400 and phase 3 is thousands. So it's a rigorous criteria, and that's where the regulation of clinical trials comes into play. And let me see, how do the students get placed at these various practices? My students, yes. We actually give the students sort of like a listing of 150 different researchers all across California, about 17 different institutions, and we give them about 3 weeks to go down that list and then choose 3 researchers with whom they interview. And after the interview, they choose one of those researchers whose research aligns with their interests, and they are happy to move geographically in that area. So it's a pretty rigorous process, because we have such a wide array of wonderful researchers who have all sorts of fantastic experience. Sometimes students come to me and they say that they want to intern like with 3 different people, because they find all the three researchers excellent, but of course you have to make a choice of one. And these 11 students have already made their choices last week, and they will be placed in the internship sites and start the internship September 1 until August of next year. And I'm seeing if, looking at the comments, thank you so, so much for all your wonderful comments towards the students and the presentations. It's much appreciated. And then there's a question by Tom, and he says, my research suggests that umbilical cord stem cells reproduce much faster and efficiently than adult human stem cells derived from skin or bone marrow, making umbilical cord cells the most powerful. Can you comment? That's a very interesting question. And umbilical cord stem cells have to reproduce faster because they are the ones which are actually providing the nutrition and all the support to the growing fetus. So from that standpoint of view, they are great. But the only flaw here, so to speak, is the number of stem cells or the quantity of the population of placental stem cells is very, very low. So 98.5% is non-stem cells. So you really have to go for that remaining, 0.8 to 1.2% and harvest those cells and expand those. And this is one of the reasons why umbilical cord is not being used as a resource for stem cells. Although despite the fact they are much faster, but in terms of harvesting, where the number and the quantity is very important, the adult tissues are preferred, such as the skin. The skin is very easy to harvest and convert them into iPSCs. And I think that's, oh yeah. And then Rick has a question. How many cell reproductive cycles are these cells capable of? Now, for a normal specialized cell, there is a limit known as Hayflick limit, and that is 52 cycles. But for the stem cells, if they remain stem cells, stem-like, there is unlimited potential. They can divide. But again, subject to the conditions being sort of appropriate for their division. Otherwise, they go into dormancy also. And if frozen, do they exist in suspended animation without multiplying? Yes, that's true. If frozen, they actually are sleeping. Let's put that way, that they actually go to sleep. And then when we warm up the tubes and put them back in their little home, which is a Petri plate, and provide them the food, they can actually survive. And this is exactly what we do. We don't keep on ordering cell lines $500 each vial every single time. We order it once and we expand it. It's like having, it's like having a huge classroom where everyone is working together. Here, we expand the cell lines and then we freeze them down. And whenever we have to start an experiment, we go to the freezer, take out a vial, warm it up, and let them grow. So that's suspended animation. And HeLa looks like it's forever. Yes, HeLa is forever because HeLa is a cancer cell. And the cancer cell is never going to die. It's just going to keep on dividing. And HeLa is one of the most notorious ones here because unfortunately, HeLa, which is cervical cancer, has an oncogene in it. And as long as the oncogene means a piece of DNA that causes cancer. And HeLa naturally has that in its DNA. So as long as that piece exists, the HeLa cell line is just going to keep on dividing forever. And what is the record for the longest line? I think, Rick, it is HeLa. I'm pretty sure. I could be wrong, but if you've read the book, The Life of Honorita Lacks, that's an excellent book. I strongly suggest everyone reads it. HeLa has grown so much for the past 55 years that if you were to collect all the HeLa cells which are grown in the world today, I think the number of cells would stretch all the way to the moon like 5 times. That's how many cells have been grown. There's some like very strange, fantastic number. I don't have the exact number, but HeLa is probably the longest. So I think we are past 12. I don't want to take too much of your time. We'll end our presentation here. So thank you so much, everyone, once again, including my students, for a wonderful presentation. And please don't hesitate to reach out to me. I'm just going to put my email here. If anyone has any questions, don't hesitate to email me and we'll be happy to answer. And yes, Rick, we can harvest the gene, but it's kind of hard because it's in the genome, but we could do it. Thank you so much for your wonderful comments. I hope you have a wonderful rest of the day. Please take care and stay safe. Bye-bye.
Thank you, everyone.
Bye. Thank you.
So long. Good luck to everybody.